today our topic is principle of inheritance, and it is a part of genetics. It could be beneficial for any standard from 9th, 10th, 11th, 12th and even to graduation level students. So here we are starting an introductory class that starts from very basics. So that is what is chromosome and how it is made up of. So here we have a cell and it contains a nucleus and a thread-like structure that is known as chromatin material. This chromatin material condenses to form chromosome. This chromosome has different parts. One strand of this chromosome is known as chromatid, while the two strands combined together are known as sister chromatid. The point where these chromatids are attached together is known as centromere. The hook-like structure present on the sides of chromosome and it occurs which help in the spindle fiber attachment during cell division and a point called locus. At locus, a gene is present, or many genes could be present on a similar locus. Terms involved in inheritance or genetics are as follow. We will start with the first one, that is inheritance. It says that there is transmission of characters from parents to progeny such that similar characters pass on from their parents to their children. Next term we have is variation. The differences between the parents and the children or among the children is known as variations. Next term we have is genetics. It is the study of inheritance and variation combined together, or in a simple language we can say that it is a study of genes. Now what are genes? Genes are the segment of DNA that code for a polypeptide. Terms are character and trait. Here character and trait, to understand its meaning, is very important because it plays a major role in genetics. An example of human being, for example, in another person we say that the person is rude, sad, angry, happy or anything that we are talking about its character based on his behavior. So here we are talking about seed shape. So seed shape basically could be of two types. It could be round or it could be wrinkled. When we specify the shape of a seed, then we are talking about its trait, not its character. For example, I have already stated about human being. Next term we have is monohybrid cross. Mono means one and hybrid means a mixture of something, or we can say that after mixing the new thing we obtain is known as hybrid. Monohybrid cross deals with only one character, whereas dihybrid cross, di means two, it deals with two characters, such that in monohybrid cross we take one character, the parents will be round, cross, wrinkled, whereas in dihybrid cross we take two characters such that seed shape or seed color, so that it could be round and yellow and wrinkled and green. Next terms involved are F1 generation and F2 generation. F1 generation is the generation obtained by crossing the two parents. The progeny we obtained is F1 generation. While after selfing the progeny or the mating the progeny with the progeny itself, the generation we obtain is known as F2 generation. Next term we have is homologous chromosome. So what are homologous chromosomes? The chromosome which are similar in shape, size, stain and everything are known as homologous chromosome. Even the gene present on the same locus or at the locus at the same point are also similar. For example, in this a gene A is present. Similarly, its homologous pair also contain a gene A dash. Here one gene represent one trait and another gene represent another trait. Are homozygous and heterozygous. Homo means similar and hetero means dissimilar. Here homo means that similar, zygous means gametes. In this case we are representing a gene by a single symbol, while we represent alleles or the pair of genes by two similar symbols. For example, in case of round I have taken it capital R, while as in case of wrinkled I have taken it small r. As both initials are same, this condition is known as homozygous condition. When I will take capital R and small r to represent a trait, then this condition will be known as heterozygous. Why? Because two different initials are present. So the gene which is able to express itself in the heterozygous condition is known as dominant gene and it is represented by a capital symbol, while the gene which is not able to express itself in the heterozygous condition is known as recessive gene and it is represented by a small symbol. Next term is test cross. When we cross the F1 generation with one of the recessive parent, this is known as test cross. When we cross the F1 generation with either of the parent, this is known as back cross. Next term is Punnett square. What does it signify? It says about the symbol or the box we use to represent the new combination in the F2 generation. Next term we have is linkage. It states that the two genes are present very close to each other such that during recombination or gametogenesis they are transferred all together. For example, here gene A and B are present together very close to each other. Therefore, whenever there is recombination, division, gametogenesis or any other process, these two genes are transferred with each other only. Therefore, the process or presence of two genes very close to each other is known as linkage, and such genes are known as linked genes. Last term we have is recombination. As we all know that in meiosis 1, prophase 1, Pachytene stage, there is a formation of chiasmatic structure or egg-shaped structure. In this structure, two homologous chromosomes or sister chromatids overlaps, therefore changing some of their genetic or chromosomal material and giving new formation. Thank you all for watching. If you like the explanation, please like and subscribe.